In 1903, an elephant named Topsy lived
Mom!
on the islands of Nova.
Tell this custard boy we gon' kill the mosquito them, you know. Boy gon' make me get Zika.
But Topsy was abused.
Malaria.
And 3 times she
Or a
fought back.
dengue.
The last time, her hands were fed her a lit cigarette. She killed him.
Bah! Tell you fi empty the drum them and kill the blasted mosquito.
One thing,
See,
Mama.
me can't even move now, me have to go hospital.
But Mama, we can't afford them
I
things,
don't
Mama.
know what I developed.
Well, Mama.
Jesus, Jesus.
This sounds like a job for the bioinformatics students!
So in order to find out if Grams really contracted the Zika, we're going to use a series of tools to test. We're going to use Mega, ORF Finder, Nucleotide Blast, Primer Blast, Electronic PCR, and NEB Cutter.
We first retrieve the Zika virus reference genome from the NCBI website. Then we extract the gene of interest using Mega. Then we use the OR Finder to find the coding sequence of the extracted sequence. Then we will use Nucleotide Blast to verify if the coding sequence is actually the gene of interest.
Well, now that we have the NS4A and the 2K genes, we can now use Permablast to identify the person. So afterwards, reverse EPCR will be used to see if the primer actually amplified the sequence from the patient. And this is basically the PCR process.
We run the sequence through the NEB cutter, we obtain a restriction enzyme to form a fingerprint pattern to identify a gene of interest, and this concludes the RFLP.
After running the protein in ORF Finder, a protein of length 537 nucleotides was identified. It was picked up in the second reading frame on the 5' to 3' strand of DNA, starting at nucleotide 23 and stopping at nucleotide 559. In order to gain primers for the process of PCR, PrimerBlast was used. 3 of the primers that were generated were tested against human genotype for amplification. 0 hits were found. This indicated that the primers would not anneal to the human genome, making them suitable for the application of PCR on a sample containing human DNA, as the human DNA would not be amplified. Finally, as mentioned before, we used the NEB cutter. BCCI was used to cut the gene. This produced 4 fragments of length 238, 159, 62, and 78 nucleotides long. If such a gene was digested with BCCI and it produced the same number of fragments of the same length, it would indicate that the gene was from the Zika virus genome. This would confirm that the person has Zika. If the same result was not yielded, it would confirm that the person does not have Zika.
After completing some of the labs, we concluded that PCR is more suitable than RFLP for developing a DNA-based test for Zika. PCR is a very sensitive, inexpensive, quite simple, and can produce results in less than an hour depending on the machine used to carry out PCR. While it may be considered as a powerful tool, it also has its limitations. This includes false positive results owing to small amounts of contaminants during the process of testing. This issue arises because of PCR's sensitivity. On the other hand, RFLP is a highly robust methodology with good transferability between laboratories and no sequence information is required. But, like PCR, there are limitations of this methodology. This includes the large amount of DNA required to carry out RFLP. Its optimization is not possible. It can be time-consuming, it is costly, and it is moderately technical demanding.
Hey, big brother.
How is this?
Okay, so we've been working on this sequence for the Zika virus, but we're actually stuck.
Alright, well, the reason why I requested this is because we're about to perform a phylogenetic test on this sequence.
What is a phylogenetic test?
Alright, my colleague will explain it for you.
Well, for infectious disease like Zika, phylogeny can help us to better understand the disease, its origin, and the agents that contribute to its spread. When we understand this, we can better handle the disease. Oh! For example, look at the Zika outbreak in Brazil and the high level of microcephaly when compared to Zika outbreaks in other parts of the world. Phylogeny can help us to understand whether this is because of a specific strain of the virus or something.
Good afternoon, everyone, and welcome to the 2016 World Health Organization ceremony. Our first presenters today will be Shamar Braithwaite and his protege, Trudian Douglas, presenting their phylogeny report on the NS4A and 2K gene. Make them welcome.
Good afternoon and welcome. If you look at the screen. First, what I'll do is I'll discuss the method my protege and I use to obtain our results, and then I'll speak on our results themselves. So firstly, we retrieve the homologues or similar sequences. And in retrieving the homologues, we looked at three things, and these are the query cover, which is how much of our original sequence align with a particular hit. We looked at the E or expected value, which is the number of hits one can expect to be seen by chance when searching a database, and an E value below 0.01 means the hit is not by chance. And lastly, we looked at the identity, which is how identical a blast hit sequence is to a particular result. So we take these homologues and we did multiple alignment on them using Mega. And then we use this multiple alignment as the input for a phylogenetic tree, which we created using bootstrapping. And what bootstrapping basically takes columns of characters from our analysis and it rebuilds a tree to test to see if the same nodes would have been recovered. So this is done over and over to give a bootstrap value. So for example, let's say we got the same node 9 out of 10 times. We get a bootstrap value of 90%, and a bootstrap value of 70% or more is accepted as good support for a node. Looking at our results, we can see that we'd have to do further testing to see if the strain to which our gene of interest belongs actually affects humans. But from what is presented here, it might not. It seems to only affect Sentinel rhesus. And from our results, we can also see that there is likely 3 specific strains of the Zika virus. So a vaccine developed for a homologue within a cluster may likely work for others within that cluster, but across clusters we'd have to do further testing to look into what happens. So that's the end of our presentation on the phylogeny. Thank you all and good night.
The different various categories of databases under which the results are presented on NCBI are genes, health, proteins, genomes, chemicals, and literature. And NCBI uses a search and retrieval system entries to search these databases. Now, most of the databases have information which may be useful in combating Zika. For example, literature has PubMed, which is a database that comprises of numerous amount of citations from Medline, Science Life Journal, etc., which all link to PubMed Central.
Now, entries on the Zika virus from relative books would aid us in finding a comprehensive description of the virus, that is, to get a better understanding of the virus. The more we know about the virus, the better able we are to deal with it. Another in genomes, which has nucleotides, which is a collection of sequences from several sources including GenBug, REFSEQ, TPA, and PDB. Genome, gene, and transcript sequence data provides the foundation for biomedical research and discovery. This database has to be very useful as it would contain information on the entire genome of the virus, which would be the foundation of the analysis of the virus.
NCBI has OR Finder, which finds coding sequences in a given sequence. However,
Transect is another useful tool that converts an entire nucleotide sequence to its corresponding amino sequence from all 6 reading frames. 6 as there is 3 on the 3 to 5 strand and 3 on the 5 to 3 strand, one corresponding to each position in the triplet codon. To select a protein using transect, you look for the start codon, the methionine, and stop at the asterisk representing the stop codon. You can confirm that this is a protein you're interested in by protein blasting the amino sequence on NCBI. Like NCBI, XPASI offers useful bioinformatics, tools, and resources. One such tool is ScanProSite, which can be used to gather information on the motif and domains of a protein. The NS4A protein was found to contain known motifs using this tool. An example of one of these motifs is the amino sequence GTL-PGH for the N-methylation site.
Another database offered by the XPASI is UniProt. UniProt offers a comprehensive resource of protein sequence and functional information. From UniProt, we found the following about our genes of interest. NS4A induces host endoplasmic regulate the ATPase activity of the NS3 helicase, while the other 2K functions as a signal peptide for NS4B and is required for the interferon antagonism activity of the latter. It was also found that an alternative name for NS4A is non-structural protein 4A, while 2K has no other name apart from peptide 2K. NS4A can be found in the host endoplasmic reticulum of the cell. A subcellular location was not given for the 2K protein. NS4A starts and ends at amino acid 2116 to 2242 respectively within the Zika polyprotein, while 2K starts and ends at 2243 to 2265 respectively. The proteins of interest does not undergo any amino acid modifications. Neither did our proteins of interest belong to any domain which is structural or contain any motif which is functional.
Our research can be used as basis for any other researcher or scientist who would like to know more about the Zika virus or have any interest specifically in the NS4A and 2K genes, its motif and domains, and other characteristics. During Jamaica's 2015 chikungunya outbreak, there were a number of deaths resulting from wrongful diagnosis and incorrect treatment. If there were to be another outbreak of a disease with symptoms similar to Zika, here or elsewhere in the world, our DNA-based test for Zika could be of use to that country's Ministry of Health. Although further testing would have to be done to test primer specificity for PCR. When devising methods of testing, our findings about PCR being more suitable for the testing of infectious diseases as opposed to RFLP could also be taken into consideration. Unfortunately, we could not conclude that it was Zika that Grams contracted. This was because the primers selected for PCR were suitable based on their GC content and lack of compatibility to the amplification of the Homo sapien genome. However, further testing needs to be done to confirm the same for the dengue and chickpea viruses before these primers can be determined optimal for the purpose of Zika diagnosis. These labs were very interesting. I hope you all learned something from us. Until next time, bye.
Bye.
Out.
Bye.
Bye.
That's all, folks.